My name is Juliana Germano. I'm a project scientist at the MAST Program. Irritable bowel syndrome, or IBS, is a chronic disorder manifested by abdominal pain and altered bowel function. IBS diarrhea represents 40% of the cases, but other subtypes also exist, and they are all a huge economic burden to the US annually. It's known that food poisoning predisposes to IBS-D, as a meta-analysis shows that IBS-D develops in 1 in every 7 cases of food poisoning. However, the pathophysiology isn't well understood. We have previously shown that CdtB anti-serum reacts with a protein in intestinal cells of Cajal and enteric neurons in rats. We have also shown that anti-CdtB precipitates a 117 kilodalton protein that was later identified as vinculin in enteric murine neuronal cell lysates. We have also shown that anti-CdtB and anti-vinculin co-localize in human ileal tissue. Moreover, we have shown that rats gavaged with C. jejuni or injected with CdtB, they show an increase in titers of anti-vinculin and also increased in water stool content. Moreover, anti-CdtB and anti-vinculin antibodies can be used to differentiate IBS-D from other GI conditions with high specificity and also positive predictive value. Moreover, in other geographic regions, other authors have also shown that IBS-D subjects, they also have increased titers for anti-vinculin and anti-CdtB antibodies. So in summary, when we have gastroenteritis from pathogenic bacteria, these bacteria, they release CdtB, and this triggers the release of anti-CdtB antibodies. But not only that, also antibodies against an autoantigen that in this case is vinculin, a cytoskeletal protein that controls focal adhesions. And we believe that this mechanism triggers post-infectious IBS. So the aim of this study was to identify and characterize the anti-vinculin antibody repertoire in IBS-D subjects to elucidate the autoimmune response and develop therapeutic strategies. A total of 13 subjects participated in the study. 3 of them were normal donors with negative titers for anti-vinculin, and 10 of them were subjects with IBS-D and positive titers for anti-vinculin. Blood was collected in EDTA tubes. They were centrifuged. B cells were enriched and activated. 10 million cells per ml were placed into OptoSelect chips that were used for antibody discovery in a beacon instrument. B-cell IgG hits for anti-vinculin and anti-CdtB were exported into 96 well plates for BCR region amplification. Later, Sanger sequencing was performed, the VJ sequence annotated, and the protein sequences were aligned. This was used for recombinant antibody production and purification into natural cells from Curia, and FACS and ELISA were used for antibody characterization for EC50s, and the Cartera LSA platform was used for capture kinetics and epitope binding. In the end, we had a list of potentially pathogenic antibodies as candidates for further investigation. This table here is to show you that IBS-D subjects, they had higher titers of anti-CdtB and anti-vinculin in the beginning of the study, and we knew that compared to the controls. And these measurements were performed by Gemelli Biotech. A total of 63 monoclonal antibodies against CdtB and or vinculin were identified by the beacon instrument. All IBS, these subjects presented positivity for anti-vinculin antibodies. These antibodies were either cross-reactive or monoreactive, while only one normal donor presented positivity for anti-vinculin antibodies, despite having very low titers by the measurements from Gemelli Biotech. Later, we used flow cytometry, ELISA, and surface plasmon resonance to characterize the recombinant antibodies. Here are examples of antibodies that were cross-reactive for vinculin and CdtB, or monoreactive for vinculin only or CdtB only, as represented by this green fluorescence. So the green fluorescence means that the B cell clones in that specific region in the sheep was producing antibodies against vinculin and or CdtB. These videos here are representing a cross-reactive antibody because we can see positivity in the same nanopens for IgG, vinculin, and CdtB. We checked the dissociation constants of the antibodies to see if they had high affinity against vinculin and CdtB, and we found high affinity for some of those antibodies, as we can see here, with some antibodies with very low dissociation constants. At the same time, we performed the FACS EC50 to see antibody potency, and we saw that EC50s were much lower for vinculin and CdtB compared to the relevant protein, meaning that these antibodies against vinculin and CdtB were very potent. We also have flow cytometry and ELISA charts here representing antibodies that were monoreactive against vinculin, as these curves in red. All curves in blue are monoreactive against CdtB, and those curves that are positive in red and blue, they represent cross-reactive antibodies such as monoclonal antibody 32. Overall, anti-vinculin and anti-CdtB antibodies were genetically diverse, as we can see here by this pairwise sequence identity heat map that was mostly blue, meaning that the pairwise identity percentage was overall low. We also checked the identity to germline, and we could see that it was varying among those antibodies, meaning that while some of them acquired somatic mutations and differ from their original clones, meaning that they were maturing, others didn't. And at the same time, we also checked the CDRH3 length, that is the length of the region that directly binds to the epitope, and that length was also changing and varying among them. But despite being genetically diverse, these antibodies, some of them, they were sharing the same epitopes, and we could see that by the cross-blocking experiment. So everything here in red means that one antibody was able to block the binding of another antibody against vinculin. And everything in green means that one antibody was able to bind to vinculin even in the presence of another antibody. When we did this cross-blocking experiment, we could categorize these antibodies into different communities. As we can see here, for example, community 1 has 4 different antibodies that were identified by the beacon instrument, and these 4 antibodies are coming from 3 different donors. If they are in the same community here, this means that they were sharing the same epitope. Then in the end, we had a phylogenetic tree with 63 monoclonal antibodies, and we could identify some of them that were characterized by different techniques, as we can see here in green. And these antibodies were also representing different epitope beams. and they will be used later for further investigation. This study had some limitations, such as the pathogenicity. We still don't know if the antibodies that were identified are really pathogenic. And due to limited sequence similarity, we may face some challenges when developing a therapy. So we conclude that antibodies to CdtB human vinculin were identified by optofluidics from all individuals with IBS-D. Cross-reactive antibodies to CdtB and vinculin supported the concept of molecular mimicry. Isolated anti-vinculin autoantibodies demonstrated sequence diversity, and despite sequence diversity, anti-vinculin autoantibodies showed potential to share epitopes. As future directions, we plan to inoculate some of these potentially pathogenic antibodies into rats to see if they develop an IBS phenotype, IBS-like phenotype. And we plan to manipulate their structure or sequence for targeted treatment. I'd like to thank the MAST program, all the participants in the study, and also Curia as our collaborator. Thank you.